Wagwan, my GCSE science students. In today's video, we're going to be looking at chapter 6 for Biology Paper 1 AQA Science Board. And in this chapter, the title is Preventing and Treating Diseases. Okay, so first up, we're going to be looking at vaccination. Then we're going to be looking at antibiotics and painkillers. Then discovering drugs, developing drugs. And then for our triple science students, we're going to be looking at making monoclonal antibodies and also how do we use monoclonal antibodies. Okay, so let's get started. Here goes. So there are some keywords that you need to know for this chapter. So one of those keywords is pathogen. Okay, and a pathogen is any microorganism which causes a disease. The next keyword which you need to know is antigen. And antigen is a substance that causes an immune response. Okay, and then you have an antibody, which is just a protein which is made by white blood cells, and that attaches itself to the antigen on the pathogen, and then eventually it kills the pathogen. Okay, so these are three keywords that you need to know. All right, so here's a look at the diagram that you guys can see here in terms of antigen and pathogen. Okay, so as you can see here, this is your pathogen. Okay, and remember what we said, the pathogen is a microorganism which causes a disease. And if you notice on the body of the pathogen, you have the antigen, and then complementary to the shape, you have the antibody, and that antibody is produced by the white blood cell, okay, which produces that antibody. Now, if you notice, different pathogens, they have different antigen shape. Okay, shapes, as you guys can see there. So therefore it means that your antibodies are complementary to the antigen, and we would need different antibodies for different antigen. Okay, so every antigen has a unique shape. The white blood cell, they produce the antibodies which shapes to fit the antigen, and the antibodies then join to the pathogen and kill them. So as we said, unique shape and therefore unique antibodies. Okay, it's almost like a lock and key, as you are seeing there. Okay, good. Let's move on. Now, why do we get ill? Now, the first time a pathogen enters the body, the white blood cell, they have to work out which antibody they're going to use. So as you guys can see here, right, you have the white blood cell here. So that's my white blood cell there. All right, it has to work out which of these antibody can bind to the pathogen. Okay, so that's your pathogen there, and this is on the pathogen you have your antigen. Okay, so the white blood cell has to work out which antibody is it going to produce. So now it's worked out. So let's go back, have a look here on the inside. Okay, good. So now it's worked out which antibody is going to produce. That one is complementary to the shape of the antigen. Okay, it's produced that one. Now that takes time. Okay, but now that it produces it, because there are different possibilities, it takes time, as we were saying. Now that it produces it, it's now able to kill the pathogen. Now, if that pathogen enters the body again, the white blood cell now has something called memory T cells. It remembers what it looks like and is able to destroy them more quickly and is produced in more abundance. And when that happens, it is said that the body now has immunity. So the white blood cells now recognize the shape for a second time as they enter. So now remember, we have these antibodies present. It remembers it. It can now produce it quickly. Okay, and therefore you don't get ill, or even if you do get ill, your body is able to fight it all quickly, and your body is now said to have immunity. Now, based on this, scientists have now come up with a method of almost quote unquote tricking the body in getting or producing these antibodies. Okay, and this is done through the use of vaccines. Now, what the vaccine does, the vaccine contains a dead, okay, very important that you know it's either dead or weakened form of the pathogen, okay, and when that pathogen enters the body, so here's your pathogen and here's your antigen. When that pathogen enters your body, the dead or weakened form, your white blood cell then decides, okay, this is the antibody that fits to it. that's complementary to the antigen and the pathogen and therefore kills it. And now, if that pathogen enters at a later point, the body will remember it. Okay, the body remembers exactly the antibodies that needs to be used, and therefore is able to fight off that disease, and therefore you're now immune. Okay, so for example, we've lived through COVID-19, so the COVID-19 vaccine would have had a dead or weakened form of the pathogen. Okay, so the white blood cells, they learn and remember how to make effective antibodies, and next time that you're exposed to it, it's able to make it quickly, specifically, and you don't get ill or as ill. Okay, good. Now, there's another term that you need to know, which is called herd immunity. Now, herd immunity is when a majority of the population, a large proportion of that population, is immune to disease. And therefore, this prevents the spread of that pathogen. Okay, so it seemingly disappears within that population. However, if the number of people who are not taking the vaccine falls, then herd immunity is lost and then the disease can reappear. Okay, for example, measles, which is happening now because people are not taking their measles vaccine, the herd immunity is lost and then we're seeing more cases of measles within that population. Okay, good. Now, antibiotics and painkillers. We definitely need to know about that. Now, an antibiotic, very important. I use the word B in antibiotic. Just to remember that antibiotic only can work on bacterial cells. So they work by interfering with the life processes such as destroying the bacterial cell wall. So antibiotics cannot work on the virus. Antibiotics only work on bacteria. Okay. Good. So they cannot kill the virus because viruses, they live within the cell. Okay. An antibiotic target our cells would be effective, would therefore be fatal. Okay. Because you'd have to kill the cells. Good. And it is difficult to develop new biotic because this takes a lot of time, one, and also that you can have antibiotic resistant bacteria, which then become resistant to the antibiotics. Okay. You also have painkillers and anti-inflammatories. Okay. So these, what they do, they treat the symptoms of the disease and make you feel better. So they're not necessarily treating what's making you ill, but they're treating you the symptoms, the effects. Okay. And you have lots of examples of painkillers. Sorry, yeah, painkillers rather, and anti-inflammatories. You have aspirin, you have paracetamol, you have ibuprofen, you have morphine. All right, all of these are painkillers or anti-inflammatories, and they don't have any effect on what's making you ill. So they don't cure it, but what they do is help you to feel better. So, for example, if you're having a headache, you take aspirin or paracetamol. It doesn't stop what's making you ill. It doesn't stop the virus. Okay, it doesn't stop the bacteria that's making you ill, only stops the symptoms. All right, so now we're going to be looking at discovering and how do they develop new drugs. Okay, so in terms of a new medicine, what are some of the things that you'd like your new medicine to do? So one, you want it to be effective, and that's a key term that you need to know. In other words, it needs to stop or prevent the disease and make you feel better. It also needs to be safe. You don't want to take something that's going to have dangerous or toxic side effects. Okay, it also needs to be stable. So over time, it must be able to be stored over or in normal conditions or in a refrigerator. Okay, and also it must be successfully taken into your body and also removed from your body. Okay, so in terms of how do we discover new drugs, or the process of how this works, is that first we have to do some sort of research and some sort of discovery. Okay, then step two, we look at then preclinical trials. This is normally done on cells and tissues and animals. Okay, good. And then we do clinical trials, which is then done on people. All right, so we need to find out, is it safe? How much do we need? Like the correct dosage and that sort of thing. So at each stage, the medicine can be rejected, and lots of time, lots of medicines are rejected even at stage one. Okay, so not a lot of medicines pass stage ones or drugs. Okay, so step one, in terms of research. All right, research, they target a disease, they identify lots and lots of new medicines. All right, that might be coming from plants, that might be coming from things such as even animals. All right, chemicals, that sort of thing. So they research, they target the disease and identify lots of possible new medicines. Okay, so as we said, traditionally they come from things like plants, mold, animal. Okay, and here are some examples. So aspirin comes from willow bark, okay, which is a plant. Penicillin was discovered from mold. All right, there's an interesting story behind that. Digitalis, which is a heart drug, that comes from foxglove, which is a plant. Okay, so that's traditionally. And now most of the drugs are synthesized inside a lab, okay, and then they extract the compound which is needed or which is taken from the plants, okay. So, which then takes us to step 2, which is your preclinical testing, okay. So here, the testing takes place in a lab, all right, and they're tested on cells, you're tested on tissues, you're tested on live animals, okay? And there's a lot of ethical issues behind this. So how do you feel about testing these drugs on animals and perhaps these animals dying over time, okay? There are lots of ethical arguments for this. However, if we don't test it on animals, are we just going to go straight ahead and test it on people? There's weighing up the pros and the cons for this, okay? So, yeah, you have to think about both sides of the argument, and that's a question that could come up on the exam, okay? The ethical argument. So they're not asking you for whether you think it's right, whether you think it's wrong, but you look at the pros, the cons, and also come up with some sort of conclusion. And then in step 3, this is now where the drug, after passing stage 1 and 2, it is now tested on human volunteers, okay? So healthy people, they're given very low doses rather, given at the start to test the side effect, to test how much is going to make it toxic, okay. And then it's also given to people who have the disease, okay. So we want to see, is it working, what dosage does it work, okay. All right, good. We want to see how effective it is, okay, the efficacy of it does it actually work, okay. Now, in step 3, sometimes this is done as a double blind trial, okay? And a double blind trial is one in which the patient don't know or the doctor don't know who is receiving the medication. And the reason that is done is so that it prevents any sort of bias. Because if you think about it, if I'm the doctor and I think that this drug is going to work, when I'm collecting my data, I'm going to be biased because I'm going to look for things which are working. And also, some patients are giving a placebo. Okay, so a placebo is a fake drug. Sometimes it could just be some sort of sugar pill. Okay, some sort of sugar pill. Okay, as you guys can see there. And the reason that we use them is because sometimes because the brain thinks they're getting something which is making you better, your brain actually tricks your body into making you think you're getting better, or actually starts showing signs of making you feel better. So the placebo is given to remove that effect. Okay, good. So this is called a double-blind trial in which neither the doctor or the patient knows who is getting the placebo, who is getting the real drug. Okay, it's all randomly assigned. All right. Now, another thing you need to know is the antibiotic efficacy, and this is for the triple science students only. So if you're doing combined science, no need to have a look at this. You could pass this part of the video, or you could stop watching the video here. So as you can see here, what we have is different antibiotics. So you have A all the way to G, and you can see what we have around it is a zone of exclusion, okay? So, and what we have here is bacteria being grown on a petri dish. Okay, so you have a petri dish, and in that petri dish you have agar gel, and that on that agar gel you have bacteria. Okay, now as you can see, there are regions where the bacteria is not growing. Now, the first question said well, let's have a look here. It says which antibiotic is most effective, and it's going to be antibiotic E. And how do I know that? Because it has the largest zone of exclusion, okay? So does this mean it has the largest zone of inhibition or exclusion, okay? That clear area, Okay, does this mean it's the best antibiotic in all circumstances? No, it does not. It means that it's really good against this particular bacteria, but could be useless against others. So for this bacteria, E is the most effective and A would be the least effective for that bacteria. Okay, good. And again, this is for the triple science students only, monoclonal antibodies. Okay, so before we have a look at monoclonal antibodies, there are some key terms that you need to know. So firstly, we need to know what's a lymphocyte. Now, a lymphocyte is an antibody producing white blood cells. So it's a white blood cell which makes an antibody, but that white blood cell, it cannot divide. Then you have a tumor cell, and a tumor cell is just a cancer cell which rapidly divides by mitosis. Okay, good. And then you have a hybridoma, which is another key term that you need to know, which is a cell which is formed when you combine a lymphocyte, which cannot divide but produces an antibody, with a tumor cell, which is really good at dividing. Together, they make what is called a hybridoma. Okay, and because of that, because you've combined them both, it is now able to produce antibodies. Okay, now when you're making monoclonal antibodies, there are some steps which you need to know. So in step one, right, the mouse is injected with a particular with a particular pathogen, and that pathogen triggers the mouse in releasing your lymphocytes. And remember, your lymphocytes, they make specific antibodies, but they do not divide. Okay, then you have your tumor cells. Okay, remember, they don't make the antibodies, but they can divide. Now you join them together to make the hybridoma. Okay, so together you now have the hybridoma, which has the antibody and the tumor cell, so it's able to divide a lot and also produce those specific antibodies. It's cloned. Now you make lots and lots of them, and then guess what? It's now able to produce lots and lots of antibodies. Now remember, these antibodies would be specific for a particular uh, pathogen or particular disease. Okay, so now that we can get them, we can separate them, we can purify them, and therefore it can be used. Okay, so to summarize, how do you make the monoclonal antibodies? You inject the mouse with a pathogen. Okay, you extract and collect the lymphocytes that make the specific antibody for that pathogen. Okay, good. After that, you combine it with the tumor cell, all right, which then makes a hybridoma. The hybridoma can then be cloned, all right, and is allowed to reproduce by mitosis. Okay, and because of that, you're now going to have lots and lots of that specific antibody which was triggered by that pathogen. Okay, and because you now have that specific antibody, you can extract it and purify it from the hybridoma. Okay, now just a quick note. So just note that that's your antigen, which is on your pathogen, which causes the disease. Okay, and then you have your antibody, which will bind to that antigen. So because of it being so specific, okay, because it's so specific and can bind to that antigen, we can then mark them. Okay, this can then be marked, and we can have several uses for these antibodies. And here are some uses. Okay, so one use is that we can use it in pregnancy tests. We could use it to diagnose diseases. Okay, because remember, the specific antibody is going to bind to a particular type of antigen which is found on certain cells. We could use it to measure and monitor certain disease. We can use it for research. We can use it for treating many diseases such as cancer. Okay, you can mark the cells for attack by phagocytes. You could block hormone. There's so many different uses because of these monoclonal antibodies. Okay, now how is it used in pregnancy tests? Now, when you're pregnant, you produce a particular hormone which is called the human chorionic gonadotropin hormone, HCG for short, okay? And this is made in the early stages in pregnancy in small amounts, and that comes out in the urine. Now, monoclonal antibodies can be produced so that they can bind to this hormone and then create that color change. So Here's your antibody, which is made by your monoclonal antibody. It's going to bind to it, to the test site, and therefore causes some sort of positive tests to show up on that. And lots of different testing works like this. Another example is the COVID test, for example, that was developed using monoclonal antibodies. Okay, so there are certain advantages that you need to know about using of monoclonal antibodies. So they can target disease or damaged cell. Okay, they can treat a wide range of conditions. Anything that can stimulate an antibody, you can treat it for that condition. Okay, it is cheaper than the other methods that's out there, but the disadvantage is that um, it possibly reduced by human cells at the starting point. So what that simply means is that you will, if you don't have in if you don't have enough of those cells at the beginning, then you won't be able to make enough of those monoclonal antibodies. All right, and making the right antibodies is quite complex. It takes time. So even though it's cheaper, it's quite complex. It takes a lot of time. All right. In terms of side effects, all right, the mouse human hybridoma, it can cause some sort of immune response, right? So you have to use a mouse human hybridoma as opposed to just the mouse ones. Okay, good. You use as little antibody as possible so they're less for the immune system to react to, because remember, these antibodies will then trigger your immune system to create some sort of reaction. Okay, and how can you improve this technique? You can do this by allowing the production of more types of antibody and attaching them to other compounds. Okay, now thank you for watching. Make sure you go over to my YouTube, make sure that you're liking, make sure that you're sharing the content with your friends, and I'm trying to get to as many subscribers as possible. Make sure you hit the subscription and share the video with your friends. Okay, I'm out.